Imagine trying to open your house door with the wrong key. No matter how hard you try, it just won't fit. That's exactly how enzymes work inside your body. Welcome to Biology Scribbles, where big concepts are explained in little doodles. Today, we're zooming into the lock and key reaction in enzymes for IGCSE and GCSE Biology. At the end of this video, you will be able to describe the lock and key model, describe enzyme, substrate, and product. Get your pen and paper. Let's dive in. So, what are enzymes? Enzymes are biological catalysts. They speed up chemical reactions without being used up. Most enzymes are proteins. Enzymes have active sites, which fits with their substrate. Hence, they are highly specific. Let's take a look at the players involved in an enzyme reaction. In every enzyme reaction, there are three main characters: the enzyme, the helper; the substrate, the molecule that reacts; and the product, the final molecule formed after the reaction. The active site. It's the most important part of the enzyme. Every enzyme has a specially shaped region called the active site. This is where the substrate binds. Just like in the diagram, the shape of the active site must be complementary with the shape of the substrate. Therefore, all enzymes can only catalyze specific substrates. Let's look into the lock and key model of enzyme reactions. The lock and key model explain why enzymes are specific. The enzyme is like a lock. The substrate is like a key. Only the correct key fits the lock. Just as illustrated in the diagram below, the reaction occurs in 3 steps. Step 1, the substrate approaches the enzyme. Step 2, it fits perfectly into the active site, forming an enzyme-substrate complex. Step 3, the reaction occurs and products are released. The enzyme is now free to work again. Let's take a look at a slightly different example of an enzyme's lock and key reaction. Here, 2 substrate molecules act as keys. Their shapes fit or are complementary with the active site of the enzyme. An enzyme-substrate complex is formed. After some time, a single product is formed and the enzyme is available to receive another substrate. The concept of denature is very important in enzyme reactions. Enzymes have active sites with a specific shape that fits with its substrate. Enzymes are proteins. The active site of enzymes may lose its shape. When this happens, the enzyme no longer fits with its substrate. Therefore, no reaction happens and no products are formed. When this happens, the enzyme is said to have denatured. High temperature and a wrong pH are known to cause enzymes to denature. In graphical representations of enzyme activity, we can always tell the enzyme has begun to denature when the temperature is past the optimum temperature or optimum pH for the particular enzyme. This is evident in the sharp drop in enzyme activity, as you can see in the graphical illustrations below. As you prepare for IGCSE and GCSE exams, remember these key terms: active site, enzyme-substrate complex, specific shape, product formed, denature. Using precise terminology earns full marks. So there you have it, lock and key model, how enzymes work. Don't forget to give this video a like and subscribe to Biology Scribbles, because here, big concepts are explained in little doodles. See you in the next lesson.